Hello there, Year 9. Here we are back on the topic B4, and today we are looking at anaerobic respiration in yeast. So, title and date please. The title is How Can We Maximise Respiration Rate in Yeast? Now today we're going to use the modelling software again, so I'll give you the link to that in the Show My Homework post. I'm also going to collect some data myself and do the graphs as I have done before, so you can use my data or you can use the program. So how can we maximise respiration rate in yeast? We're on lesson 11, and make sure you've done the date please. If you need extra time, then pause now. So anaerobic respiration in yeast. Anaerobic respiration in plant and yeast cells is represented by this equation. Anaerobic because there is that lack of oxygen, so it is simply glucose, just as it would be if it was in human cells. And with that yeast, it goes through this process called fermentation, and it doesn't make the lactic acid that it would in a human. It makes ethanol and carbon dioxide, and that ethanol can be used in the production of alcoholic drinks, wines, beers and spirits, and the carbon dioxide is used in bread to make bread rise. Okay, so the carbon dioxide bubbles up and causes the bread to rise. So we do need to write some notes from this page please. So anaerobic respiration in yeast cells is called fermentation, has economic importance in the manufacture of bread and alcoholic drinks. So we put yeast into our bread, and the yeast anaerobically respire, producing that carbon dioxide causes the bread to rise, and the yeast in the production of alcohol forms that ethanol. Okay, so we're going to write those notes, and if you need extra time on that page, then pause now. Okay, so this is the program that we're going to use. We're going to measure the rate of anaerobic respiration in yeast, and the way that we're going to measure that is to measure the amount of carbon dioxide produced in a fixed period of time. Okay, so we've got this inverted burette. You'll find these in chemistry. Okay, so our scale is going to be slightly upside down, and the carbon dioxide is going to be formed in this conical flask with the yeast in there, the glucose in there. We can change the temperature, and the carbon dioxide will feed through this delivery tube, and we will measure it here. Okay. Before we can start, we need to get a results table. Now, normally I would do this investigating temperature and glucose as two separate experiments. I thought that might be a bit drawn out today, so I'm just looking at how temperature affects the rate of anaerobic respiration in that yeast. So we're just going to get one set of results rather than three sets and calculating means, so it should be quite quick. If you want to, you can have a look at glucose and how that affects the rate of anaerobic respiration as well, but that is totally optional and you can collect those results yourself. So this looks quite similar to other experiments we've done in this topic. We've got our results table there, and we're going to copy that down, and if you need extra time, then please pause now. So here is the program. I'm just grabbing a pen and paper so that I can write down the results as we go. And you can see I've got different glucose concentrations. I can change that here. I'm going to keep that where it was set. Remember, I said you could change that as a second independent variable should you choose to as a separate experiment. I'm going to go for a reaction time of 10 minutes. Because that's what I put into my table, and I'm going to look at different temperatures starting at, I can't remember what was on the table, so I'm going to start at 5 degrees C, working my way up to probably about 60 degrees C. I probably should have looked to see what was on my table of results, but never mind, hopefully I'll collect enough results. Okay, so I'm going to go play, and you can see the bubbles filling up in the burette here, although we have no bubbles being produced. That must be a fault in the model there. They've produced bubbles even to show that the experiment's working, I suppose, but we haven't actually collected any. So at 5 degrees C we had no gas produced. Then I'm going to go and click back, and get that up to 10. Hard to see on this scale, but if you look up here, you can see more carefully where you've got to. So 10 degrees, after 10 minutes, we're on 0. And let's go now to 15 degrees, and again, and still 0. Not very exciting at the moment, unfortunately. And let's go up to 20. And we've finally got some gas produced. Now be careful how you read this because it's kind of upside down, okay? 0 is here and 1 is here. And it's where the middle part, the meniscus of that bubble is. So I've got that at 20 degrees, 0.5. Okay. And now we'll go to 25. Much more gas produced this time, and I'm saying that is 3.9. And let's go for 30 degrees. 30 degrees C, I should say. And it's kind of halfway between 13.9 and 14. I think I'm going to go for 13.9. Okay, and moving up to 35 degrees C. Now we started talking about enzymes before, and we've said that enzymes like this sort of temperature, so we should get a nice big score, and there we have. So at 35 it was 29.8. And then they don't like to get too hot because they start to denature. We kind of touched upon that word but not talked about it too much. So we've got 37.5, our biggest score so far. And I imagine, if I'm going to do some predicting, that it should drop now, going to 45 degrees C. And it has to 29.2. Okay, let's jump up to 50 degrees C now. And we've got 22.4. Again, I'll keep going. I can't remember what I took that results table to. I should have looked more carefully, shouldn't I? 55 degrees C, and we've got 9.3. So definitely dropping now. And let's go to 60 degrees C, and we've got 1.9. I think I might just go until we get to 0. So 65 and only 0.2, and then 70. Pretty sure it's all denatured by this point. Yes, 70 is no gas produced. Okay, let's go put those into the results table. Okay, here are the results that I've collected, and I have put them into the table. You can see I have actually included another couple of little lines at the bottom there with the results for 60 degrees C and 65 degrees C. I just wanted to finish it off really just to show how much it drops and how quickly it drops when you increase that temperature up. So I had collected enough data for the table that I had, and I have extended that table just slightly there. Okay, now you can do a graph on graph paper, or you can go to Excel as I have been doing before, and that's where I'm going now. If you need extra time to collect these results and write them in your table, then pause now. So as you can see, I've put all of my data into Excel now, and I'm just going to highlight that data that I want to put into my graph, and I'm going to insert that graph. And I've got continuous data, so I'm going to go for a line graph. So here it is, and although I can't do my lines of best fit, I actually think that's quite a nice curve of best fit there, so I'm quite pleased with that. I'm going to add those chart titles. So the horizontal is the temperature, and insert symbol. Let's find that degree sign, there we go. degrees C. Okay, and then I'm going to go back to the design and add a vertical column, vertical label, length of carbon dioxide, oops, bubble produced in 10 mins, and that is in centimetres. I can't make that 3. Should be a 3 like this one, and I can't do that because my my menu items have disappeared in this format, so I'm just going to go for a 3. Wouldn't normally do that, but does the job for today. Okay, and I'm going to lose that. Now sometimes when you do this, if I put maybe a higher concentration of glucose, some of those readings would have gone beyond the burette. So, you know, it would have filled the burette and more. So it's useful to try and, you know, choose variables. I think in the past where we were looking at limiting factors in photosynthesis, we actually always got the optimum. But sometimes these programs aren't, they're flawed a little bit in that they go beyond the readings. That's why I didn't get the a higher concentration of glucose. I was afraid that I'd fill the burette as I got to my optimum temperature. So we can see the optimum temperature here is 40. I'm going to take that and put that into the PowerPoint to get the results, to look at those results and do some analysis. Okay. If you need an extra time, pause now. Okay, so here we've got our graph in situ with our questions. So we've collected the data, we've plotted the graph. We can identify the independent variable as the temperature in degrees C. The dependent variable was the length of carbon dioxide bubble produced in 10 minutes. And that was our indicator of the rate of anaerobic respiration in the yeast. And describe the relationship at the start. We can see that at the start, as the temperature increases, so does the length of carbon dioxide bubble there. So therefore, we can say as temperature increases, the rate of reaction increases. And then suggest what happens later. Beyond the optimum point or the optimum temperature of 40 degrees C, we can see that we get this dropping, and that's because the enzymes are denaturing, and therefore it's basically killing off the yeast. So when you put your loaf of bread in, the yeast allows the bread to rise, and then the yeast is destroyed by the end of the cooking process. Okay, so that gives us our kind of analysis of that graph. If you need any extra time to write some notes, then pause now. And then let's look at our last part of this lesson today. A quick 4 marker. Yeast is used in the brewing and baking industries. Why is yeast used in these industries? Okay, so this is a question that just kind of pulls together all the ideas that we've come up with today. So we talked about alcoholic drinks, we talked about bread making. Okay, why is yeast used in these industries? What is it that is produced that helps those industries, the brewing and the baking industries? Have a little think, give yourself 4 minutes, write down 4 points of information. If you need extra time, then pause now. Okay, well done. Hopefully you've given that a good go, and let's have a look at our answers. So we've got the fact that carbon dioxide is produced for 1 mark, ethanol is produced for another mark, and then you say how they're used. So the carbon dioxide is to make bread rise. The carbon dioxide also makes the alcoholic drinks that are fizzy, like the beers and the ciders, to fizz. And the ethanol is the alcohol present in those alcoholic drinks, for example, beer, cider, wine and spirits. Okay, so that gives us our 4 marks. So there we are, we've come to the end of this lesson. Hope you found that interesting. Hope you enjoyed doing some practical work in some form and drawing that graph and analysing it. And I will see you again next lesson. Thank you.